In the previous video we have studied about the benefit of mycorrhizas. So if you haven't watched the video, you can go to the channel and watch the video. Today we are going to study about isolation of AM fungi. The first method of isolating of AM fungi is wet sieving method. Here what happens that this wet sieving method is also known as decanting method, given by Gerdman and Nicholson in 1963, developed to isolate different size of spores. Here this wet sieving method was introduced, and the process was to isolate different size of spores. The soil near the root system is collected, and an aqueous suspension is passed through different sieve to collect spores of different size. The spores of the fungi is collected by the near of the soil, and an aqueous suspension is passed through and it is a process through which the spores has to pass, and the different sizes of spores are collected in the sieves. The wet sieving and decanting is one of the popular technique when compared to other techniques. It is the most popular technique if compared to other techniques. This technique is used for sieving the coarse particles of the soil and retaining AMF spores and organic particles. Sieves of different sizes means that here the coarse particles of the soil and retaining AMF spores means here the technique for sieving is for the coarse particle of the soil, and from that coarse particles of the soil, we are getting, we are retaining AMF spores and organic particles from the soil, that coarse particles of soil, by the process of sieving technique. Now the process of technique, how the process take place, phase by phase or step by step. Now we will read here. 10 gram of soil was mixed with 100 ml of water in the 500 ml conical flask. Means in a 500 ml of conical flask, we take 100 ml of water, and 10 gram of soil, and we will mix it together. The soil mixture was agitated vigorously to free the AMF spores from soil and allowed to settle for 15 to 45 minutes, and the supernatant was decanted through standard sieve. Means that after we take conical flask of 500 ml, and we put 100 ml of water and 10 gram of soil in the conical flask, we will mix it, and after mixing vigorously, means with full force, we will mix it very fast, and after mixing it, we will see that AMF spores are free from the soil particles, and we will let it allow to settle down the spores in the bottom of the conical flask for 15 to 45 minutes. We will give rest to the conical flask to 15 to 45 minutes, and the supernatant, the liquid left above the mixture of soil and water, we will decant that water through standard sieve by using a dissecting microscope. Now we will take a dissecting microscope. Spores were picked by means of peptide or needle. We will put the spores underneath the microscope, the dissecting microscope, and we will pick up the spores to separate it. We will pick up by the with the help of pipette and the needle. Earlier, Gradman in 1955 devised the first useful technique for extracting spores from the soil. A soil sample was suspended in 4 times its volume of water. Heavier particles were allowed to settle for a few seconds. Then the liquid was decanted through a sieve with 1 mm mesh. Means that the mixture of the soil particles and the mycorrhiza and the water, if the mixture, we will what we will do that the suspended in 4 times volume of water, the heavy particles of the uh, spores, that fungi spores will settle down in few seconds, and the liquid left we can sieve it through a 1 mm of mesh. Mesh is a net like cloth through which we will pour the water in the another container. Whatever passed through this sieve was then poured through another sieve with 0.25 mm mesh. Now the leftover material we get in the container, the second container. Now we will again pour this this mixture of water, we say, in another through another mess, which will have 0.25 of the space in the net, in the mesh. Material spores picked out by hand under a dissecting microscope. Now material spores will be picked out by hand under dissecting microscope. A technique given by Gerdman 1955 was slightly refined by Gradman and Nicholson in 1963, who used the following series of sieve: 1.0 mm, 7110 micrometer, 420 micrometer, 250 micrometer, 105 micrometer, 74 micrometer, and 44 micrometer. They found that most of the desired spores fell in the 420 to 149 micrometer range. The most desired spores, the means desired spores mean the spores that we want for the further production or further process. The most desired spores fell in 420, between 420 to 149 micrometer ranges, and they use this fraction for their study, and they use these fractions, means these spores, and these fractions for their further studies. Now sieving method: a soil sample plus sterile water. We will take soil sample and sterile water in the conical flask, the it and mix it, and there will be hot water. Filter and sieve, we will mix it and filter, and we will sieve it. 719 micrometer to 250 micrometer, 50 micrometer, and 45 micrometer. We will sieve it through these all the through all these micrometers mesh. Now spores separated from soil particles. Spores will be separated from the soil particles, mixed with carrier material, use when required as fertilizer. Now after the separation of the spores from the soil particles, we will mix it with the carrier material to keep it for the further process. Then we will use when it is required for the biofertilizer. Now here is the diagrammatic: wash soil sample with water. We will wash the soil sample with water. Now pour it into the 750 micrometer sieve. Here it is, sieve is present. We are pouring this mixture in the 750 micrometer sieve. Now again we will pour it to the 250 micrometer sieve, then to 150 micrometer sieve, and then third 50 micrometer sieve. Collect, then add water and centrifuge 5 minutes at 2000 rpm. Means that now after all this process, we will collect the leftover material, and we will add water and centrifuge for 5 minutes in 2000 rpm. Now here we can see three test tubes with resuspended pellets in 50% sucrose and centrifuge for 1 minute to 2000 rpm. Here what is discharge floating debris with supernatant. Here the discharge debris are floating with the supernatant. Now supernatant to sucrose. Here the material is supernatant to supernatant washed on 50 micrometer sieve to sucrose before filtration. Before filtration we will wash it to the 50 micrometer sieve. Here this material is discharge pellet. Now this is the filter paper or other filtration method. By this method we will filter and collect the spore collected and kept in petri dish. Spore will be collected. Now we will keep the spores in the petri dish. Here the petri dish is this container, it is petri dish and this, and here the spores are present in the petri dish. This whole process is a wet saving process. Now the next process, sucrose gradient method, we will do it in the next video.